Hello class 11th, a very good morning to all of you. So our today's topic is chemiosmotic synthesis. As you know that we are finished with our light reaction, and light reaction includes, that is the cyclic phase as well as non-cyclic phase of photophosphorylation. So during this phase we have seen that there is a formation of ATP. How this ATP during light reaction is synthesized, that is explained and understand. We are going to understand that thing with the help of chemiosmotic synthesis. So chemiosmotic synthesis or chemiosmotic is a mechanism which help us to understand ATP synthesis, or you can just say that ATP formation, that is going to be understood by the chemiosmotic synthesis. Now as you can see a diagrammatic view here to explain chemiosmotic synthesis, we are going to explain this diagram, but I will request you to listen to me first one step by step, and then I'm going to explain the theoretical part of this hypothesis. Now we have taken here, this structure is a thylakoid. So we are going to, I have taken the individual thylakoid. So this thylakoid has two membrane. One is the, as you can see, this is the outer membrane, outer thylakoid membrane, and on the inner side, towards the inner side, there is a inner thylakoid membrane. So two membranes are clear, outer thylakoid membrane and inner thylakoid membrane. Now towards the inner side of the thylakoid membrane, there is a complex present for the splitting of water, or you can say the complex is for the photolysis of water. What is photolysis? In which water is going to break down, or water is going to release, that is electrons, protons and oxygen as the byproduct. So where does this splitting of water or photolysis of water takes place, or where is the complex present? That is towards the inner membrane of thylakoid. Now, or towards the outer side, or on the outer side towards the, I will say specifically stroma, there is a present enzyme, that is NADP reductase enzyme. Now I use the word stroma. Where is this thylakoid present? This thylakoid is present in the stroma. As you can see these green dots, that is our stroma, and towards the stroma on the outer side of the membrane, there is a present enzyme which is NADP reductase enzyme. Then there is present two proteins. There is a new word for you, F0 particles or F1 particles. Now just towards the inner side, or in between the outer and inner membrane, there is a present integral protein. There is a present integral. What is the integral? Another name, that is the intrinsic proteins. Intrinsic proteins or extrinsic proteins, they are present in the plasma membrane. So here also there is a integral or intrinsic protein which is called F0. It is present towards inner side. On the outer side there is a presence of F1 particle or F1 protein, which has ATPase enzyme which help in synthesis of ATP. Clear? So I talked about two particles, that is F0 and F1 particles. F1 is the integral and F1 is the extrinsic proteins. So these are basically channels. These are the transmembrane or the channel proteins which are going to help us in this chemiosmotic synthesis or in the ATP synthesis. Now we have discussed photosystem 2 and photosystem 1. Once we talk about photosystem 2, non-cyclic photophosphorylation, because this in this diagram we cannot draw each and everything in detail, so we have just mentioned here photosystem 2. You know that what photosystem 2 or non-cyclic photophosphorylation has, that it accept the electrons they receive, absorb the light, then they get excited. Here is the primary electron acceptor. It will transport the pair of electron, or it will transfer the pair of electron. So here in the purple color I have written here the electron transport chain. Electron we have just shown here it as a electron transport chain, and the another side there these electrons they are passed on to photosystem 1, right? These things you have already done in the non-cyclic photophosphorylation. As you can see now, that once the photosystem to absorb light at a particular wavelength, the electrons they get excited, and these electrons they are passed on to electron transport chain. अब electron transport chain हम पूरा तो नहीं draw कर सकते हैं, like PQ, CY, PC. So here I have taken only the cytochrome complex. Now once the electron they move from plastoquinone to CY, that is your cytochrome complex, these cytochrome they release some amount of energy. Now that some amount of energy that is used to pump the protons. This is which side it is? This is stroma side of the thylakoid. So this is stroma basically. So these protons from the stroma they are received by cytochrome. Now cytochrome already have pair of electron. These electron pairs they are transferred to now next part of the next member of the electron transport chain. Once it pass the electron to the next member of electron transport chain, the protons they are pumped into the lumen. Lumen is the inner side area of the thylakoid. This is outer, this is inner membrane, and inner space is called that is lumen, or it is a cavity, or it is a basically empty space. So protons that is your H positive they are pumped to now lumen. ये कहाँ पे आ गए हैं? ल्यूमेन में. सो इलेक्ट्रॉन साइटोक्रोम क्या करता है? प्रोटॉन को एक्सेप्ट करता है, और जैसे ही इलेक्ट्रॉन ट्रांसफ़र होते हैं नेक्स्ट to the next member of ETC, then it release these protons into the ल्यूमेन. तो ये प्रोटॉन्स कहाँ आके इकट्ठे हो गए? ल्यूमेन में. नाउ ऑन द अदर साइड वाटर इज गेटिंग splitted, and water is getting broken down. Now after water is getting broken down, the water will release a pair of electron. These pair of electrons they will be received by photosystem 2, and then it release a pair of protons as well as oxygen. Oxygen it is released as the byproduct. But what happened to these protons? These protons again now they are coming to the lumen. So lumen has now more number of protons, means inner side of the thylakoid has more number of protons as compared to the outer side. और जब कभी भी concentration में difference होता है, that create a kind of a gradient. Gradient means difference in the concentration. Here proton they are different in their concentration. Outer side they are less, and inner side or in the lumen they are more. So now they have to move from higher concentration to the lower concentration. It means they need to move from stroma to need to move from lumen to the stroma. So how they will move? Now these protons they are coming with the energy. These protons they will coming with the energy. They will be received by here F0 particles. What are F0 particles? They are the integral proteins present in the plasma membrane. So now these proteins, they receive this protons, they are coming with energy. Now once these protons they enter into F1, F1 क्या करता है, F1 इनकी एनर्जी को यूज़ करता है, which in through which ADP combine with inorganic phosphate. Now once ADP combine with inorganic phosphate in the presence of enzyme, there is a formation of ATP, and that is how these protons they released into the stroma. F1 ke through now they come through the stroma. Now here as you can see, electrons they will be travel to the photosystem 1. photosystem 1 से excite होके वहाँ पे primary electron acceptor present है, वो receive करेगा. Now on the outer membrane there is a present NADP. NADP के साथ में प्रेजेंट है यहाँ पे NADP रिडक्टेज एंजाइम. सो यहाँ पे जो प्रोटॉन स्ट्रोमा में गए हैं, दे विल मूव टू दीज NADP, दे कंबाइन विद दिस NADP एंड फॉर्म NADPH2 इन द प्रेजेंस ऑफ एंजाइम NADP रिडक्टेज. सो जैसे ही ये इलेक्ट्रॉन ट्रांसफ़र होते हैं NADP के पास, these protons they join with NADP in the presence of enzyme NADP reductase. There is a formation of NADPH2. If I ask you what are the two sources of release of protons in the inside the lumen, सबसे पहला तो है इलेक्ट्रॉन ट्रांसफ़र. इलेक्ट्रॉन्स दे आर ट्रांसफर्ड थ्रू द इलेक्ट्रॉन ट्रांसपोर्ट चेन. दैट इज़ द फर्स्ट सोर्स ऑफ protons in the lumen. The another is the splitting of water. Then the major thing you have to remember here is that is F0 and F0. They help in the synthesis of ATP. So this is a diagrammatic explanation of chemiosmotic synthesis. That is what you have to draw in your exam. Now what you have to write down in theoretical portion, two ways. First thing, you have to describe the two pathways or two methods through which protons, that is your H positive, they are accumulating. They starting accumulating in the lumen. So first one is photolysis of water, or you can say splitting of water. When second is, I already told you, when electrons they moving through ETC, means we are talking about here this ETC chain. So when electrons they are moving through ETC, that is electron transport chain, energy is used. एनर्जी इज़ यूज क्योंकि यहाँ पे सपोज हमारे यहाँ पे फोटो सिस्टम 2 है. It will pass the electron to electron transport chain, which is your cytochrome complex. Now once electrons they move through electron transport chain, there is energy is released. This energy is used. यहाँ से जो energy during electron transfer energy release होती है cytochrome के पास, जब ये electron पहुँचते हैं तो energy is released. This energy is used to accept the electrons from stroma to the lumen. So electrons from stroma and then pump the to accept the protons. Electrons from the stroma and pump the protons into the lumen. Basically, that is here you have to write down. That is the yes, don't get confused with this, that electrons they are accepted from the stroma. Electrons they are coming from the stroma, so don't get confused here that how electrons they are coming from the stroma, because electrons they are coming from the photosystem 2, but protons when these electrons they are transported, energy is used to pump the protons into the lumen, which create high concentration of protons. So F0, F0, then here is a present your F0 and then is the F1. This act as a channels, or you can say helpers, which help the electrons to move from lumen into the stroma. Now, chemiosmotic synthesis is explained. It explained the mechanism of ATP synthesis. We are already clear with that. ATP synthesis is directly linked to agar proton gradient. If proton gradient will not be developed, there will never be transfer of the protons. So across the membrane of thylakoid, one must condition is, that is the proton gradient. And proton gradient kahan se aayega? First one is the splitting of water, and second one is, now this there is the membrane, and PEA is, here is the PEA. What is PEA? PEA, that is the primary electron acceptor. So as you can see that this located towards the outer side of membrane, this is the outer side of membrane, and now it transfer its electron to the cytochrome, that is your hydrogen carrier. So cytochrome here, cytochrome is electron transfer also, as well, or you can say electron transporter also, as well as it is hydrogen, or I will use the term proton carrier also, right? So here we are talking about cytochrome. Now cytochrome complex, it removes a proton from the stroma. जब वो ट्रांसपोर्ट करता है इलेक्ट्रॉन, तब ये रिमूव कर लेता है एक्स्ट्रोमा, एंड जैसे ही वो नेक्स्ट मेंबर को देता है, इट रिलीजेस द प्रोटोन ऑन द इनर साइड ऑफ द मेम्ब्रेन. जस्ट अंडरस्टैंड दिस केयरफुली दैट हियर is your present photosystem 2. Now it is passing the electron to the electron transport chain where cytochrome is present. During the ट्रांसफ़र ऑफ इलेक्ट्रॉन, और इलेक्ट्रॉन जैसे ही यहाँ पे साइटोक्रोम के पास पहुँचते हैं, एनर्जी इज़ रिलीज. दिस एनर्जी इज़ यूज टू एक्सेप्ट द proton. Now this cytochrome pass the electron to the next member of ETC. Then it release the proton into the lumen. So when it releases the proton on the inner side of membrane, कब release करता है? When it pass its electron to the electron carrier on the inner side or the lumen side of the membrane. Now the enzyme NADP reductase कहाँ पे present है? It is present on the outer side of the membrane towards the stroma. And जैसे ही electrons यहाँ पे पहुँचते हैं PS1 के पास, it protons यहाँ पे जैसे ही PS1 के पास इलेक्ट्रॉन पहुँचते हैं, स्ट्रोमा में जो आपके प्रोटॉन्स हैं वो रिलीज़ हो जाते हैं. नाउ दीज रिलीज्ड प्रोटॉन्स दे रीच टू द NADP पॉजिटिव, एंड देन NADP पॉजिटिव के पास पहुंचने के बाद NADP पॉजिटिव रिडक्टेज एंजाइम गेट एक्टिवेटेड विच कन्वर्ट NADP टू NADPH2. आई थिंक दिस वन इज़ क्लियर. जैसे ही यहाँ पे क्या प्रेजेंट है हमारा ETC विल गिव द इलेक्ट्रॉन टू PS1 अल्टीमेटली गिव द इलेक्ट्रॉन टू NADP. जैसे ही ये इलेक्ट्रॉन NADP के पास आ जाते हैं, देयर इज़ द रिलीज ऑफ stroma, or you can say once the electron they reach to PS1, there is a release of protons in the stroma, and these protons they join with NADP. So integral proteins, they are present in thylakoid, which are F0 particles, and extrinsic protein, they are F1. F1 act as an enzyme ATPase, and ATPase help in synthesis of ATP. So when protons, now they are यहाँ पे बहुत सारे प्रोटॉन्स हमारे इकट्ठा हो गए हैं. व्हेन दीज प्रोटॉन्स दे हैव टू, दिस इज ल्यूमेन. नाउ दीज प्रोटॉन हैव टू मूव इनटू स्ट्रोमा में जाना है इनको. सो वंस दे मूव फ्रॉम ल्यूमेन टू स्ट्रोमा, यहाँ पे कौन उनको हेल्प करता है? सबसे पहली हेल्प उनको मिलती है F0 पार्टिकल्स इन द फैसिलिटेटेड डिफ्यूजन। फैसिलिटेटेड डिफ्यूजन हम जानते हैं कि वो डिफ्यूजन जिसमें हमें हेल्पर की ज़रूरत होती है। तो यहाँ पे ट्रांसपोर्टेशन के टाइम पे कौन फैसिलिटेट कर रहा है? F0 पार्टिकल्स। नाउ protons, they are coming with energy, so with energy they enter into F1 particles and this energy used by F1. अब F1 क्या कर रहा है? This is F0 and this is F1. F1 is using this energy to combine ADP with the inorganic phosphate and form the ATP. So that is how your ATP is synthesized at every step of light reaction. So what you need to remember, that first thing for ATP synthesis, proton gradient is needed. It is need to be generated. वाटर स्प्लिटिंग कॉम्प्लेक्स के लिए साइट क्या है? इनर मेम्ब्रेन। NADP आउटर मेम्ब्रेन पे प्रेजेंट है। ऑक्सीजन कहाँ प्रोड्यूस हो रही है? ल्यूमेन में। ATP और NADPH2 प्रोडक्शन कहाँ हो रही है? स्ट्रोमा में। बहुत बार मैंने ये चीज़ें अब रिपीट कर दी हैं। नाउ द ग्रेडियंट इज़ ब्रोकन डाउन। जैसे ही ये मूवमेंट होती है प्रोटॉन्स की अक्रॉस द चैनल, मीन्स there is a movement of these protons to these F0 F0 protein, that is your channel proteins. So they consist of enzyme ATP. So ATP has two parts, F0 F1. So there is the breakdown of gradient. It provide enough energy to cause a structural conformational is the structural change. और जैसे ही F1 receive करता है H positive, there is a conformational change and ATP is get activated and ATP molecules are synthesized. Now what do you need for a chemiosmotic synthesis? A membrane. Which kind of membrane? A thylakoid membrane. You need a proton pump. One side there should be higher concentration and another side there is a low. Only then that proton gradient is created. Proton pump means basically how these protons they are added into the thylakoid. One is with the splitting of water and another one is through the electron transport chain during the electron transfer basically, and then you need a ATPase. Energy, see, every time energy is used. Once protons they are first time coming from stroma and received by CY, then energy is used because यहाँ पे energy release होती है, राइट। साइटोक्रोम कॉम्प्लेक्स जब इलेक्ट्रॉन के पास, साइटोक्रोम के पास जब इलेक्ट्रॉन्स पहुंचते हैं, then there is a energy is released. That energy is used to pump the protons from stroma to the lumen. So one, that time energy is used. Second energy, so it is used to create a gradient or high concentration of protons within the lumen. जिस इस एनर्जी की वजह से ही there is a accumulation of protons inside the lumen. So that you have to write down in the chemiosmotic synthesis. One point here again I will repeat, that don't get confused with this point, that when electrons they are moving through ETC. Now see here he is talking about the first non-cyclic. Non-cyclic में you know that photosystem 2, that will be release electron which will be received by primary electron acceptor. They will pass through the electron transport chain. Now when these electrons they reach to the cytochrome complex, energy is used to accept the electron. Now once there is a release of energy, जैसे ही यहाँ पे इलेक्ट्रॉन पहुँचते हैं साइटोक्रोम के पास, energy is released. This energy will accept the proton from stroma. Now this will cytochrome pass the electron to the next membrane, that is PC. Then that time it will release these protons into the lumen. राइट, सो डोंट गेट कन्फ्यूज्ड. और जैसे ही ल्यूमेन में ये रिलीज करेगा, देयर विल बी द हाई कॉन्सेंट्रेशन ऑफ प्रोटॉन्स इनटू द ल्यूमेन, क्योंकि एक तो इसने रिलीज कर दिया और सेकंड हमारा स्प्लिटिंग ऑफ वाटर में रिलीज हो गए. सो देयर विल बी नाउ high concentration of protons into the lumen. This is all about chemiosmotic synthesis. So I will meet you in my next lecture with the dark reaction. Till then, go through all the phases of light reaction and take care of yourself. So I will meet you next time with the